Okay, welcome to the discussion on the lab concerning the cardiovascular system, the heart, and the blood vessels. The objectives for the lab that we went through last week are shown here. Remember that you want to be able to identify not just the dissected sheep heart, the human heart models, but also be able to identify the heart muscle tissue um, on a slide or on a PowerPoint, be able to identify the intercalated disc, um, and be able to distinguish an artery from a vein. And we're also going to discuss the pathway of blood through the heart and the pulmonary and the systemic circulation. So what you're looking at here is simply the heart as it exists inside the thoracic cavity. Now, it sits in an area between the lungs called the mediastinum, and within the mediastinum, which uh, we will show you by circling here, okay, this is the mediastinum, okay, the heart sits inside a special chamber called the pericardium, which we will circle here in blue, okay, that is the pericardium right there. Pericardium means around the heart. On either side of the mediastinum sits the pleural cavities. We'll draw the pleural cavities here in red. Okay, this is the right pleural cavity. Okay, and this is the left pleural cavity. And they're all encased inside the bony thorax, which is surrounded by the rib cages laterally, anteriorly, and posteriorly. The ribs articulate with the vertebral column at posterior midline and with the sternum at anterior midline. The heart and the lungs lie right above the diaphragm, this muscle right here, which is the main muscle of breathing. And every organ inside the thoracic cavity sits in a double layered sac. The lungs sit inside a pleural sac. There is a parietal pleura that actually clings to the thoracic inside lining of the thoracic cavity, and there's a visceral pleura that actually clings to the lungs. The heart sits inside the pericardial cavity, which is made up of the parietal pericardium, which is attached to this fibrous outer layer here. In between, there is a space, pericardial space that contains a slippery fluid called pericardial fluid, and then there's a visceral pericardium that is right next to the organ. And so the purpose of all these membranes is that they produce a lubricating fluid that allows the internal organs to move without generating a lot of inflammation, irritation, and scarring, which would end up impeding the function of the organ. Okay. The apex of the heart is this area up here to which the great vessels are attached. The great vessels include the superior vena cava, the inferior vena cava, which is behind the heart and we can't see, the aorta, and the pulmonary trunk. Okay, the pulmonary trunk is the gateway to the lungs from the heart for the blood, and the aorta is the gateway to the body from the heart for the blood. The superior and inferior vena cava bring the blood back to the heart from the body, and what we can't see, which we will see in the next slide, are the pulmonary veins which bring the blood back to the heart from the lungs. And you can see a little better here in the heart model each of these different structures. So if we were to just look at the external anatomy of the heart, we start with this model showing you this is the front of the heart. So this is the right side of the heart, and this is the left side of the heart. The two upper chambers of the heart are called the atria. There is the right and the left atrium. The two lower chambers of the heart are called the ventricles. There is the right ventricle and there is the left ventricle. The right and left atrium are separated by an intraatrial septum that keeps the two chambers separate, and the right and left ventricle are separated by an interventricular septum that keeps the two lower chambers separate. The atria receive blood. Okay, the right atria receives blood from the body through the superior and inferior vena cava. The left atrium receives blood from the lungs through the right and left pulmonary veins. The ventricles only receive blood from the atria. What happens when the heart pumps is that it will flow from the upper chambers to the lower chambers through what we call atrioventricular valves. The right atrioventricular valve is called the tricuspid, so named because it has 3 flaps, and the left atrioventricular valve is called the bicuspid or the mitral valve, so named because it has 2 flaps. The veins, the vessels that lead out of the heart include the pulmonary trunk, which takes the blood from the right ventricle to the lungs, okay, and the aorta, which takes the blood out of the left ventricle to the body. All right, so that is the front of the heart. The back of the heart, you can see here, shows you some important features. Now we can see the inferior and superior vena cava, which bring oxygen poor blood back from the body to the heart. And you can see here the pulmonary veins, which bring blood back from the lungs to the heart. Now you might say, now wait a minute here. I notice that these veins are red instead of blue. And what you have to understand about arteries and veins is that they're not identified as a result of the color of the blood that flows through them. They're identified strictly as a result of the direction of blood either to or from the heart. So remember this, always, 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 okay? And we'll use black as our color here. Arteries, okay, always carry blood away from the heart, away, okay? And veins always carry blood back to the heart, regardless of the color. Okay, we'll get into why the colors are what they are when we talk a little bit more about the cardiac cycle. The left atrium is shown here, receiving blood from the pulmonary veins. You can see the left ventricle down here. This is the descending aorta, which will pass out of the aortic arch right here and will eventually run down behind the heart. It'll eventually go through the diaphragm, where it'll then be called the abdominal aorta, and then eventually we'll get all the way down to the base of the abdominal aorta, where the vessel will split into the right and left common iliac arteries, which will then branch into the internal and external iliacs. The external iliac will then become the femoral artery as it passes into the leg, just to give you an idea of some of the overall route that the blood takes through the body. But we'll get into more detail about that when we actually look at a map of the major blood vessels in the human body. For now, I just want you to understand how the blood is flowing through these vessels. Okay, it's coming back to the heart from the body through the superior and inferior vena cava, shown here, okay, and it's also coming back to the heart through the pulmonary veins, okay, through here. Okay, so blood's flowing in this way, and blood is flowing out this way, okay, through the pulmonary arteries and through the aorta, okay, so out that way. Okay, and this is the back of the heart. This is the posterior view. Okay. It's very important that we also understand that the heart is itself an organ, and so it needs a constant blood supply. It receives its major blood supply from the coronary circulation. 2 of the major vessels that supply the coronary circulation include the right and the left coronary arteries, which branch off the base of the aorta, which is the gateway to the body, okay, for oxygen-rich blood. And again, you can see the other great vessels here. One of the things that we should touch on before we move to the next model picture is a look at some of the external features of the different chambers of the heart, I want you to notice that the upper chambers, the atria, have relatively thin walls, okay, but they are relatively expandable walls as well. Okay, this is why sometimes these flaps are called auricles, because they look a little bit like ear flaps, and what happens is as they fill with blood, they bulge outward. And then I want you to look at the lower chambers here, the ventricles, and notice how much more muscular they are, and that's because they have to do more work than the atria do in moving blood around the body. Okay, the right atria, as we're going to find out, is going to pump blood to the lungs. The right ventricle is going to pump blood to the lungs. The left ventricle, as we're going to find out, is going to pump blood to the aorta, and from there it'll head out to the rest of the body. Okay, so the blood enters the atria, falls from the atria to the ventricles, and then the ventricles discharge the blood to the lungs in the case of the pulmonary circuit, and to the body in the case of the systemic circuit. Now, if we open the heart up and look inside it, what we see is some of the internal features that make the heart function as an efficient one-way pump. You can see here the openings of the right and the left coronary artery at the base of the aorta, again supplying oxygen and nutrients to the heart muscle itself. But as we remove the front of the heart, we've taken basically a transverse section through it. You can see here that the upper chambers, the atria, are far less muscular than the lower chambers are. The lower chambers have much thicker walls because they do more work. The thing I want you to notice also is that of the four chambers, the left ventricle has the thickest wall because it does the most work, okay, because it has to pump blood to the entire body, and as a result it has to overcome a much more significant amount of back pressure force than the right ventricle does, since it only has to pump blood to the lungs, and there's a lot less resistance in that circuit. You can also see the atrioventricular valves and some of the semilunar valves in this picture. This is the right atrioventricular valve, known as the tricuspid, because it has three flaps. Notice that the flaps are attached to the base of the ventricle by these little tendons, which are called chordae tendineae, and what those tendons do is they keep the flaps from flipping back in the other direction towards the atria when the ventricles begin to squeeze. So this is part of what ensures that the valves that open from the atria to the ventricles only open in one direction, which is from the top chamber into the lower chamber, and never in the reverse direction. Okay, the bicuspid or mitral valve has a similar setup. It has two flaps instead of three, hence the name bicuspid, and it is also tethered by the chordae tendineae to muscles in the base of the ventricle called the papillary muscles, and again this keeps the flaps from everting. One of the things that we're also going to find out as the cardiac cycle proceeds is that the papillary muscles exert a slight amount of force on these tendons that opens these flaps and allows the blood to spill from the upper to the lower chambers in order to complete now the cardiac cycle. So they do give a little bit of a tug in order to make sure that these flaps stay open when the blood is spilling from the top of the heart down into the bottom. The apex of the heart, shown here, is inferior to the base, which is where the great vessels attach. You can also see one of the two semilunar valves. This is called the pulmonary semilunar valve. It is the gateway to the lungs for the blood that's coming out of the right ventricle. And then sort of behind here, it's a little bit hidden. You'd have to rotate the heart a little bit to see it. At the base of the aorta is the aortic semilunar valve, which is the gateway to the rest of the body for the blood that's coming out of the left ventricle. Okay. Now these valves are also one-way valves. They only open into the great vessels. Okay, so the pulmonary semilunar valve opens into the pulmonary trunk, and the aortic semilunar valve opens into the aorta. And then when there's pressure in the opposite direction, what will happen is that these flaps will shut and the blood will not be able to re-enter the ventricles the same way that when the ventricles begin to exert pressure, these atrioventricular valves will shut and the blood will not be able to re-enter the atria. Okay, so you can just see here in this diagram the setup for the coronary arteries. You can see them branching off the base of the aorta here to supply the heart muscle proper. Okay. Now, what we're looking at here is just sort of a diagram showing you how it is that the heart tissue gets perfused. Essentially, the aorta forces the blood into the coronary arteries. It then feeds the myocardium, and then as the oxygen is used in the blood that perfuses the myocardium, the oxygenated blood will eventually drain back into the right atrium as it enters the coronary sinus, and then it'll eventually be pumped into the right ventricle. From there, it will be pumped into the pulmonary trunk, and from the pulmonary trunk, it'll eventually make its way to the lungs, where it will then become reoxygenated. Now, occasionally what can happen is that the coronary circulation can become blocked. In the event that that happens, the heart muscle itself will be starved for oxygen and nutrients, and if this occurs, heart muscle tissue can die. And of course, heart muscle tissue, um, after it suffers a trauma like that, does not recover very well. It does not repair or divide to any great extent. So once we damage the heart as a result of, say, a heart attack or of impeded circulation in the heart muscle, the heart is weaker from that point on. So that's a very important point to try and hammer home here. One of the ways we get around these blockages, if we have to repair them, is that we can insert an inflatable device via a catheter, and this is a procedure called a balloon angioplasty, where essentially we inflate this balloon and we open this vessel up by forcing the plaque against the walls and restore circulation to the heart muscle proper. This often happens as a result of a lifetime of having a diet that's very high in cholesterol and saturated fat. The deposits on the walls of the arteries eventually can occlude them, and this can result in a lack of perfusion to downstream tissue. If it happens in the heart, it can cause a heart attack. If it happens in the brain, it can cause a stroke. Okay, what you're looking at here is just a diagram showing you the flow of blood through the heart. Okay, a nice little chart showing you the blood delivered to the particular structure. Okay, and the valve that it passes through. Okay, and what we'll do is we'll go through each of these components when we talk about the cardiac cycle here in just a moment. Okay, so this is a nice chart to kind of help you keep track, but what we'll do is we'll address the cardiac cycle in detail in just a minute. We touched on the fact that the heart is found inside the pericardial sac. The inside wall is lined by the parietal pericardium. You can see it here. There's a fibrous layer that anchors it to the surrounding tissue. And then the inside wall is called the visceral pericardium, and there's pericardial fluid in the cavity between these two layers called the pericardial cavity. Beneath the visceral pericardium, which can also be called the epicardium, we find the myocardium. This is where the heart muscle cells lie. And then you've got the endocardium, which is essentially endothelial tissue that lines the chambers of the heart and repels the formed elements so that we don't engage in damage to the formed elements, which could result in spontaneous clotting of the blood. Okay, again, the fluid here is to lubricate the heart as it beats so that it doesn't damage itself every time that it moves. This is an organ that's always in motion, so it needs a lot of lubrication in order to compensate for that ringing motion that the heart engages in every time that it pumps. Now this is a sheep heart, and we're looking at the front of the sheep heart here, and you can see here a tag in the right atrium, the right ventricle, the left atrium and left ventricle. There's the apex of the heart. You can see here the interventricular sulcus. This is the dividing line between the right and left ventricle here. You can see the left auricle here and the right auricle up here, the little flaps that are allowing the upper chambers of the heart to expand when they fill with blood. This is just a zoom up of the anterior surface of the sheet heart, and what we're doing here is we're inserting a probe into the pulmonary trunk, which comes directly off of the right ventricle. Okay, this is one of the ways that you can tell which vessel is which. Remember that the pulmonary trunk is always going to be the frontmost vessel. The aorta is going to be right behind it, okay, and it's going to have a relatively thick wall. Okay? Remember again, of course, that the apex of the heart is the inferior portion, the base is the superior portion. What we've done here is we've just put some more probes in the heart. We're looking at the back of the heart now. You can see the aorta here, okay. This is the pulmonary trunk coming off the right ventricle. This is the aorta coming off of the left ventricle. You can see here the left atrium, the left ventricle down here, okay, and the right ventricle, which would be on the other side. Okay, so remember, we're looking here at the back of the heart, the back of the heart. Okay. If we were to take a transverse section through the heart, what we would find is that the wall of the left ventricle is considerably thicker than the wall of the right ventricle. And the reason for this is that the left ventricle has to pump blood to the entire body, while the right ventricle only has to pump blood back and forth to the lungs. And remember that the lungs are not very far distant from where the heart is, right? The lungs lie on either side of the heart in the thoracic cavity, and so there's less resistance for that chamber to overcome in order to move the blood out. The left ventricle, however, has to overcome considerable resistance in order to pump the blood to the rest of the body. That being said, always remember that the same volume of blood is pumped by the right and left ventricle in each cardiac cycle. It's just that the power that's exerted by the right ventricle is less than the power exerted by the left ventricle because of the difference in resistance that those chambers have to work against. Okay, you can see the interventricular septum here as well. If we want to name some of these vessels, there's the superior vena cava, the aorta, there's the pulmonary trunk, okay? There is the right atrium, there is the left atrium, there is the pulmonary vein, okay? And what you're looking at here is just a peek at some of the structures you're going to need to identify on the heart model or on a PowerPoint. So if we were to trace the flow of blood through the heart, we can do that now, and let's use, let's try, so we'll try, what would magenta look like? Let's try, we'll try, well, let's start with blue, okay? So deoxygenated blood returns to the heart through the superior vena cava here, okay, into the right atrium, and through the inferior vena cava here into the right atrium, okay? But it also comes through the coronary sinus into the right atrium, okay, and enters from the back side of the heart, okay? So there's a tiny pit or an opening that the blood from the coronary sinus flows through and enters from the heart muscle itself. So the right atrium is getting deoxygenated blood really from three directions. The next thing that happens is as this chamber begins to fill with blood, the pressure begins to build, and as the pressure is higher in the upper chamber than the lower chamber, what will happen is that the valve between the upper and lower chamber will open up, and the blood will spill from the right atrium down here into the right ventricle. Okay, you can see it right here. Okay, blue for deoxygenated blood. And then what will happen is that the right ventricle will begin to squeeze, and as it does, this valve up here will close and will prevent the backflow of blood in the opposite direction. Okay, so the blood is not going to go back into the atria. As the pressure continues to build, eventually what will happen is that the pulmonary semilunar valve will open up and the blood will exit through the pulmonary semilunar valve into the pulmonary trunk and will flow from the pulmonary trunk into the pulmonary arteries towards the lungs. Okay, so this is the blood flowing through the pulmonary arteries towards the lungs. Okay, again, the blood has a low oxygen concentration, so it's going to appear bluish through the walls of the vessels through which they flow. Okay, now what happens in the lungs is that you breathe, and the blood that's coming to the lungs from the heart will pick up oxygen and will change color from a dull to a very bright red. What will then happen is that the blood will then drain into the pulmonary veins, and from the pulmonary veins we will drain into this chamber, which is the left atrium. Okay, so you can see it right here. Okay, blood draining in. Okay, it's going to go into the left atrium. Okay, so just sort of visualize this coming through this side here. Okay, as this oxygenated blood fills the left atrium, eventually what's going to happen is that it's going to spill from the left atrium into the left ventricle. The left ventricle will begin to exert pressure, and then this atrioventricular valve, the bicuspid, will close, and then the blood will continue to build pressure, and eventually it'll pop this vessel open, which is the aortic semilunar valve, and then the blood will flow through the aorta, and from there go to the rest of the body. And we'll indicate this in yellow because it's a little tough to see here. Okay, there's the blood flowing through the aortic semilunar valve. Okay, and then from here into the aorta, and from the aorta it's going to go to the entire body. Okay, all over the body. Okay, so that's the path of blood flow through the heart. Now, what I want to make sure that you understand is that as the heart beats, all four chambers are always going to have some fluid in them at all times in order for the heart to be a functioning pump. That's what we call priming the pump. Okay, and so if we're looking at a cardiac cycle just in one cycle's worth of movement, what we find is that the two upper chambers will squeeze first, forcing blood into the two lower chambers. The two lower chambers will then squeeze next, and they'll force blood into the pulmonary semilunar valve, into the pulmonary trunk in the case of the right ventricle, and through the aortic semilunar valve into the aorta into the case of the left ventricle. So when the heart's beating, it's these two chambers squeeze, and then these two chambers squeeze, and these two chambers squeeze, and then these two chambers squeeze. Okay, so pump, 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 pump. Okay, always. And that's what's responsible for the sounds that the heart makes, is the opening and closing of these valves. As they open and shut, you get the typical heart sounds that we call lub and dub. Okay, you can see here the interventricular septum separating the two lower chambers of the heart, the atrioventricular septum that separates the atria from the ventricles, and behind these great vessels the intraatrial septum that separates the two atria from each other. Here again, of course, is the tricuspid valve, the bicuspid valve, the aortic semilunar valve, the pulmonary semilunar valve, and the aortic semilunar valve. Now, if we look at a sheep heart, this is looking at it from the front. This is a coronal section taken through the sheep heart. You can see the tricuspid and bicuspid valves here with the pin stuck in them. This is the interventricular septum. This is the wall of the left ventricle. Note how thick it is. Okay, This is the left ventricle itself. Okay, you can see the right ventricle as well. Okay, and up here you can see the right atrium and the left atrium here, indicated by the letter F. Okay, remember the flow of blood right from the right atrium through the tricuspid valve to the right ventricle, and on the other side from the left atrium through the bicuspid valve to the left ventricle. If we turn the heart on its side, you can see a few more of the features. Again, you can see the right atrium, the right ventricle down here. Okay, this is the tricuspid valve. Here you can see the aorta. Okay, over here you can see down here at A the left ventricle. These are the chordae tendineae, which are attaching to the bicuspid valve here, which is the left atrioventricular valve, and then you can see right here the aortic semilunar valve as well. Okay, so this is the heart cut in transverse section. This is a little bit simpler view. You can see the aortic semilunar valve here, and you can see the interventricular septum. In this case, we're looking at the back of the heart. Okay, this is the right ventricle. This is the left ventricle here. Okay, so this is the other half of the heart that was missing from this side. Okay. All right. So if we look at a single cardiac cycle, what they're showing you in this diagram is how the blood flows through the heart. So diastole is what's described as the heart chamber relaxed and basically expanding to its maximum volume. So that's when the muscles in the heart wall are not generating any significant amount of force. Systole is described as the heart chamber squeezing, and so this is when the heart muscle begins to contract, and eventually what this will result in is the movement of blood through the chamber. So during early diastole, the ventricles are filled by the atria passively. Then the atria will contract to fill the remaining blood from the upper to the lower chambers in order to top off the ventricles as they fully expand and fill with blood. The next thing that will happen is that the ventricles will begin to contract, the atrioventricular valves will shut, and then as the pressure continues to build, as systole continues in the ventricles, eventually the semilunar valves, the pulmonary semilunar valve in the case of the right ventricle and the aortic semilunar valve in the case of the left ventricle, will open and the blood will be expelled into the great vessels, and from there we will head to the lungs in the case of the pulmonary trunk, and we will head to the body in the case of the aorta. Okay. And so this is how the blood begins to fill. Okay, there's diastole, the blood's entering, the ventricles then begin to fill, then they squeeze. Okay, the pressure builds, and then eventually what happens is the pressure builds sufficiently is that we pop these doors open, and we're able then to force the blood into the great vessels and towards their destination. Now, one of the things that will happen after the blood is forced into the great vessels is that these pulmonary and aortic semilunar valves will shut. The blood will then flow backwards briefly, but it won't re-enter the ventricles because the valves will prevent that, and that actually produces one of the heart sounds, okay, the second heart sound. Okay, so you can see here just a look at how the heart is functioning. You can see here in mid to late diastole as the ventricles begin to fill, okay, the blood spilling from the upper to the lower chambers. And then what happens is that the ventricles begin to squeeze. As they squeeze and produce force, okay, and while both the doorway into the ventricle and the doorway out of the ventricle are closed, what's happening is that the force is building, but the volume of the blood in the chambers is not changing, and so we call this isovolumetric contraction. As the pressure continues to build and the exit to the ventricles opens up, we call this ventricular ejection, and that's how the blood gets into the great vessels. And then what will happen is that the chambers will relax, okay, all four chambers will expand to their full volume. This is called isovolumetric relaxation because the amount of blood in the chambers isn't changing while the volume of the chambers is expanding. Okay, and so that's what we call early diastole. So we start with uh, mid to late diastole, then ventricular systole, then early diastole, and that helps draw the blood in. Okay, and then we start the entire cycle over again. Now, if we look at the electrical activity of the heart, which coordinates this beating, one of the things that we see is that in order for the heart to beat as a coordinated unit, we have to have the signal spread in a uniform fashion from a particular origin across the walls of the atria, down the interventricular septum to the apex, and then up the walls of the ventricles. And then following that signal, that particular chamber will squeeze. Okay, so the pacemaker of the heart is called the SA or sinoatrial node, and it initiates the signal because the cells that make up the sinoatrial node have what we call an unstable membrane potential due to the fact that the membrane itself is leaky. Okay, so ions tend to drift into the cytoplasm, and eventually what happens is that the membrane will reach what we call a threshold voltage, and that will result now in the production of the electrical signal. And this wave of depolarization will then flow from the SA node to the AV node, or atrioventricular node, which has the nickname the electrical gateway to the ventricles. The signal will then pause there for a very brief period to give the lower chambers a chance to fill with blood, okay? And then what will happen is that the signal will rapidly shoot down the interventricular septum through the bundle of His and into the right and left bundle branches. From there we get to the apex, okay, of the heart, and then we'll radiate up the walls of the ventricles through the Purkinje fibers, and then the ventricles will squeeze. Okay, so at the beginning of systole, the tricuspid and bicuspid valves close. The heart valves close loudly and create the lub heart sound. At the end of systole, the dup heart sound is caused by the closing of the semilunar valves. Okay, so the atrioventricular valves closing, the semilunar valves closing, that's what's responsible for the heart sounds that you hear in a typical heartbeat. Okay, and so you can see here where this happens on an actual EKG. Okay, it happens at the peak of the QRS complex for lub, and it happens at the shoulder of the T wave for the dub. And so here you can see how the heart sounds work into the contraction phase. Okay, there's lub as these valves shut, right, because of the pressure building up on them and closing them. And here is dub as the semilunar valves shut due to the backflow of blood against them. All right, so the cardiac cycle right there. Now, what you're looking at here is just how the electrical signal moves through the heart in order to produce the heart cycle. Okay, so it starts with the sinoatrial node initiating the electrical signal that spreads across the walls of the atria. What follows is that the atria will contract. They'll go into systole and inject the rest of the blood into the ventricles. While the signal waits at the atrioventricular node for the ventricles to fill, it will then be shot down through the bundle of His into the bundle branches, down to the apex, and then it will radiate up the walls of the ventricles, and that will be followed by ventricular systole. Okay? So most of the time when you're sleeping, eating, sitting, studying, voiding, and defecating, the heart rate is going to be relatively normal, about 75 beats per minute. And then for a short period of time, if you say increase your activity level, or when you're under some other sort of exertion, what will happen is that you will have sympathetic innervation from the autonomic nervous system that will cause the pacemaker of the heart to speed up a bit, and the heart rate will begin to rise. Okay, so you can see here how the decreasing heart rate is what we experience most of the time. Okay, the increasing heart rate is what we experience when we increase our activity levels or when we sense a threat. Okay, and all of this is governed by the sympathetic and parasympathetic divisions of the autonomic nervous system, affecting the way that the SA and the AV node move the signal through the heart. So what you're looking at here are just some of the factors that tend to accelerate heart rate and increase cardiac output. Cardiac output is defined as the heart rate times the stroke volume. For instance, if you watch a scary movie or you're almost in a car accident, you're going to increase your cardiac output by increasing your heart rate while the stroke volume remains the same. If you're in an accident and you suffer trauma and you begin to hemorrhage, you'll increase the cardiac output by keeping the heart rate steady and increasing the stroke volume. So you'll make the heart squeeze harder as it ejects the blood. So there's more volume per pump, even though the number of pumps per minute isn't changing. Exercise does both. It'll increase the heart rate and increase the stroke volume, and the result is that the cardiac output will go up considerably. The cardiovascular system is a closed system. The blood stays either in the heart or in the blood vessels. Okay, it's not supposed to leak. Leaks generally indicate disease or trauma. And there's two major circuits: the pulmonary circuit, which is between the lungs and the heart, and the systemic circuit, which is between the body and the heart. And then there's a minor circuit, which is from the heart muscle back to the heart. So that goes from the coronary arteries to the myocardium back to the right atrium. Blood that exits the heart passes through an artery, and blood that comes back to the heart comes through a vein. And so what you're seeing here in this diagram is just a schematic that illustrates how the blood moves through this system. Okay, so as blood drains back to the heart from the body, it enters the right atrium through the inferior and superior vena cava. As this chamber fills with blood and the pressure goes up, the pressure in the ventricle stays the same. The blood will spill from the upper to the lower chamber. Then the lower chamber will enter systole. As the pressure goes up, this atrioventricular valve will close. The pressure continues to build. Eventually, the pulmonary semilunar valve will be forced open. Okay, and the blood will then exit through the pulmonary trunk and head to the lungs. In the lungs, it'll pick up oxygen as a result of the diffusion of oxygen into the blood from the air that you breathe. At the same time, carbon dioxide will be removed from the blood as it diffuses out of the blood and into the air that you exhale. And then the blood will change color to a bright red. This blood will then flow back to the heart through the left and right pulmonary veins into the left atrium. From the left atrium, as the pressure builds, eventually what will happen is that the bicuspid valve will be forced open. The blood will spill from the upper to the lower chambers, assisted by atrial systole. And then the last part is when the left ventricle squeezes, forcing the bicuspid valve shut. The pressure continues to build. The aortic semilunar valve will open, and the blood will flow out of the heart through the aorta, and from there, oxygenated and nutrient-rich blood will deliver these life-sustaining components to the tissues of the body in order to keep you alive. What you're looking at here is just a comparison of the anatomy of an artery and a vein. Notice what they look like in an actual section of tissue. The arteries tend to be rounder and have a thicker middle layer called the tunica media, which is made up primarily of smooth muscle, while the veins tend to have an irregularly shaped lumen with much thinner tunica media, and they also have valves to ensure the one-way flow of blood through them as the muscles around them squeeze the blood through the valves one at a time, and eventually it'll reach the heart and then will re-enter uh, the circulation, will re-enter the cardiac cycle, and will be pumped back to the lungs, and then the whole cycle will repeat itself again. Um, the tiniest and most delicate blood vessels in the body are the capillaries. They simply have a lining of endothelial cells, and they're designed primarily for gas transport. And the reason being that we don't want to have a significant barrier between the blood and the tissue fluid and the surrounding tissue once we get to the capillary bed, so that we can deliver nutrients to the tissues and remove waste products, and also deliver oxygen to the tissues and remove carbon dioxide. And then eventually what will happen is that this oxygen poor, nutrient poor blood will then flow back into the venous return, and from there eventually we'll head back to the heart. Okay. The same three tunics are present in arteries and veins: the tunica intima, which is endothelial tissue; the tunica media, which is smooth muscle; and the tunica externa, which is connective tissue. It's just that the thicknesses of the different tunics are different when you compare arteries to veins. And also remember that veins have these valves that the arteries lack. Because the arteries carry blood under higher pressure, they have to have a thicker wall. That's why the more extensive tunica media and the rounder shape of the lumen. The smooth muscle in the vessels helps to maintain blood pressure and to redirect blood flow. When the tunica media contracts and the lumen of the vessel shrinks, that's what we call vasoconstriction. When the smooth muscle relaxes and the tunica media allows the lumen to open up, we call that vasodilation. When a vessel is experiencing vasodilation, the blood pressure in it will drop. When a vessel is experiencing vasoconstriction, the blood pressure in it will rise. In addition, we can also redirect blood by squeezing down on some vessels and opening up others in order to get blood where it's most needed in the body from moment to moment. At any one time, only about a quarter of the capillary beds in the body are fully perfused, and what they do is they switch off in order to maintain oxygen and nutrient delivery to our tissues. If we look at the pressure changes and the overall pressure in the vessels in the circulatory system, what we find is that the further away we get from the heart, the lower the pressure becomes, and the lower the pressure differential becomes. And the reason for this is that we are further away from the driving force. In addition to that, we're experiencing force in the opposite direction, which we call drag, which essentially dampens the force against the walls, and also dampens the difference between the systolic and the diastolic pressure. And the systolic pressure being when the ventricles squeeze, the diastolic pressure being present when the ventricles are relaxed. Okay, so the further we get away from the heart, the further we are from the force, the more drag we experience, the lower the overall pressure becomes, the lower the difference between the two pressures becomes. Once we get to the arterioles and to the capillaries, the pressure drops dramatically, and the reason for this is that these vessels are so small that a significant amount of resistance is experienced. As a result, there's a big dampening effect on the overall pressure in the vessels. As we head into the venules and the veins and eventually back to the vena cava, the pressure again is minimal. One of the things that we should note is the fact that when we look at the design of the human body, veins tend to be closer to the surface than arteries, with just a very few exceptions, okay? Those being the pulse points of the body. And the advantage of this is that if we are injured, we have a laceration or a puncture, and we open up a vein, we're going to lose a lot less blood than if we open up an artery. If we nick an artery, you'll never forget it, because the blood will come out under extremely high pressure. It'll hit the ceiling in spurts. So we tend to protect the arteries a little bit more than we do the veins. If we look here at the blood flow through capillary beds, what we find is that they're designed for gas exchange. They have very thin walls, and so they allow the diffusion of gases, nutrients, and waste products down their concentration gradients in order to constantly deliver oxygen and nutrients to the tissues that need it to live, and remove waste products so that they don't accumulate and change the conditions around tissues such as pH and electrolyte concentration to the point where chemical processes stop. So when tissues need oxygen, when they're hypoxic, what will happen is that the capillary bed in the systemic circulation will perfuse. So what will happen is that they will vasodilate. These sphincters, which are doorways into the capillaries, which are also made of smooth muscle, will open up, and what we'll do is we'll flush the area, we'll flush the capillaries that run through that area with a lot of blood. When the tissues do not require oxygen, what will happen is that the sphincters will choke down, the amount of perfusion will drop until such time as the tissues generate a low oxygen condition again. Okay, so this is what we mean when we say that at any one time about a quarter of your capillary beds are wide open and about 3/4 are shut. And this is A, to maintain blood pressure, and B, to alternate which tissues are receiving oxygen and nutrients from moment to moment. If we were ever to undergo a physiological change, it would open all of these capillary beds up. The drop in blood pressure would be so dramatic that we would pass out. Tissues also require fluid because this is what helps maintain their temperature and pH, and it also provides a medium in which chemical reactions can occur. Blood pressure forces water into the tissue at the arterial ends of the capillaries, and water tends to move out of the tissues at the venous ends of the capillaries. In addition, we should note that at the arterial end of the capillary, oxygen and nutrients are delivered to tissues, and at the venous end of the capillary, waste products and carbon dioxide are removed. Okay, and this is all again due strictly to concentration gradients. Okay, this is how it is that we deliver the nutrients to keep the tissues alive and remove the waste products so that it don't build up to toxic levels. Okay. What you're looking at here again is just the same diagram we saw earlier. Under low pressure, the blood has to get back to the heart. In order to do this, we take advantage of an anatomical feature in the venous system, which are these one-way valves that allow the blood to be forced by skeletal muscle in one direction back towards the heart from areas that are below it. In addition to that, the fact that you breathe, and when you breathe, that produces pressure changes on the vena cava. That helps to milk the blood back up towards the heart from areas below it, because again here we're fighting gravity. Areas above the heart don't have as great a challenge because they're working with gravity. There are still valves there, and the skeletal muscle activity still does help, but again the major challenge is here from areas that are below the heart. Okay, we end up here looking at a comparison of skeletal, cardiac, and smooth muscle. Just remember that in cardiac muscle we have the intercalated discs. Cardiac muscle tends to have one nucleus per cell. It is striated, meaning that there is an overlap of actin and myosin in a regular fashion, forming what we call the sarcomere. And remember that these intercalated discs are there for two reasons. They hold the heart muscle cells tightly together so that they don't come apart when the heart contracts, and they also provide a method to move charged particles between cells that are adjacent through tunnels that are called gap junctions. Okay, so they're both what we call a communicating junction and a mechanical junction at the same time. The smooth muscle tissue and the skeletal muscle tissue lack this. They look very different under the scope. This is actual micrographs of the different tissue types. You can also see differences in shape. Skeletal muscle tissue tends to be elongated and has more than one nucleus per cell. cardiac muscle tissue, more branched with one nucleus per cell. Both of these are striated. You can see the vertical dark and light lines moving through the fibers. Smooth muscle is totally different. The cells tend to be spindle shaped and have no striations. Remember that cardiac and smooth muscle are not under voluntary control, while skeletal muscle is. Okay, we end up with just a diagram showing you a little bit more clearly the blood flow through the heart. Okay, so if you can label this diagram, you're in pretty good shape with regards to the cardiac cycle. You can see here just another view of the heart. This is out of your lab book. Again, be able to label all these major vessels. We will practice this, okay? And we're going to have probably some slides of the sheep heart when we take the upcoming practical on this particular chapter. So remember to be able to identify the chambers in the valves in the sheep heart, as well as in a diagram or on an actual model. Okay, so I will end this by showing you some videos on the cardiac cycle and how the electrical signal moves through the heart, and then I'll see everybody in class next week. Okay, thank you very much.
Fresh air entering the lungs carries oxygen to the alveoli. The amount of gas in air or of gas dissolved in a fluid can be expressed as partial pressure, which is measured in millimeters of mercury. The partial pressure of oxygen in the air within the alveoli is 104 millimeters of mercury. Carbon dioxide that enters the alveoli from the blood causes the carbon dioxide concentration in the air of the alveoli to be 40 millimeters of mercury. Carbon dioxide is continually removed from the alveoli as air is expired. Blood coming from the heart at the arterial ends of the pulmonary capillaries has a partial pressure of oxygen, or PO2, of 40 and a PCO2 of 45. Therefore, oxygen diffuses from the air in the alveoli into the blood, and carbon dioxide diffuses from the blood into the alveoli because of these differences in partial pressures. Oxygen and carbon dioxide diffuse until there is no difference in partial pressure in the air and the blood. At this point, there is no more net movement of O2 or CO2. Therefore, at the venous ends of the pulmonary capillaries, the PO2 in the blood and alveoli is 104, and the PCO2 in the blood and alveoli is 40. Some oxygen was removed from the blood to nourish lung tissue. Therefore, the PO2 in arterial blood leaving the heart is 95. Oxygen diffuses out of the arterial ends of tissue capillaries into the tissue fluid, then into the cells, and carbon dioxide diffuses out of the cells into the tissue fluid, then into the blood because of differences in partial pressures. At the venous ends of tissue capillaries, the PO2 in the blood is equal to the PO2 in the tissue fluid, and the PCO2 in the blood is equal to the PCO2 in the tissue fluid, resulting in no more net movement of O2 or CO2. The blood now carries the O2 and CO2 to the lungs. In the body, all of these exchanges occur simultaneously.
Ventricular contraction causes the atrioventricular valves to close, which signals the beginning of ventricular systole. The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open. When the ventricles relax and their pressures drop, Blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole. Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles. This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling. Action potentials originate in the sinoatrial node and travel across the wall of the atrium from the sinoatrial node to the atrioventricular node. Action potentials pass slowly through the atrioventricular node to give the atria time to contract. They then pass rapidly along the atrioventricular bundle, which extends from the atrioventricular node through the fibrous skeleton into the interventricular septum. The atrioventricular bundle divides into right and left bundle branches, and action potentials descend rapidly to the apex of each ventricle along the bundle branches. Action potentials are carried by the Purkinje fibers from the bundle branches to the ventricular walls. The rapid conduction from the atrioventricular bundle to the ends of the Purkinje fibers allows the ventricular muscle cells to contract in unison, providing a strong contraction.